Hello students, welcome back to KTHM e-learning portal. Myself Shazeen Khan from Department of Botany, KTHM College, Nasik. And today we are to discuss the topic of secondary metabolites from the subject of biochemistry, credit 4, and the chapter name is phytochemistry. We will be studying the topic of secondary metabolites in today's session. In the last lecture, in the previous session, we have discussed in detail about the primary metabolites, how they are synthesized, how they function in the regular growth and development of organisms and plants. And we have seen the different precursors of primary metabolites, how they synthesize, the different pathways used for their synthesis. In today's lecture, we will be seeing the introduction of secondary metabolites. What are secondary metabolites? Then we will be seeing how primary metabolites, they act as precursors of secondary metabolites. And also, we will be seeing the different classes of secondary metabolites, the structure and properties of the common secondary metabolites. Let us start with the introduction. Now, plants produce a large, diverse array of organic compounds, and some of them they have a direct function in the growth and development, while other group of the chemical compounds they have no role or they have no direct role in growth and development of plant. These chemicals, which I am telling about, that they are not having any direct role in growth and development of plants, these chemicals or these substances, they are known as secondary metabolites, secondary products or natural products. So secondary metabolites, they are produced by the plants, but they do not have any direct role or they are not involved in any function of growth and development. The secondary metabolites, as I said earlier, they have no recognized or direct role in the processes of the growth and development. Now, what the different processes are there for the growth and development? For example, photosynthesis, respiration, solute transport, translocation, protein synthesis, nutrient assimilation, differentiation, or the formation of carbohydrates, proteins, and lipids. So, in all these functions or all these processes which are listed above, the secondary metabolites, they have no direct role in these important life processes. Now, secondary metabolites, they also differ from primary metabolites in having a restricted distribution in plant kingdom. Now, the primary metabolites, they will be found in almost all the plants, but the secondary metabolites, they will be found in a certain group of plants, or a single secondary metabolite will be found in a particular plant, not all. So that is why they are different in distribution from the primary metabolites. And also a particular secondary metabolite, they are found in a related group of plant species, whereas primary metabolites, they are found throughout the plant kingdom. In comparison with primary metabolites, which are essential to plant growth and development, secondary metabolites have internal roles in plants and they are useful to the plants with respect to certain interactions with the environment or communication of a plant with its environment. The different interactions with secondary metabolites result with the plants. Now, the different interactions can be as follows. The secondary metabolites can be involved in the accumulation of pigments in flower petals. Also, some secondary metabolites may be released by the plants uh, to attract the flower, to attract the pollinators. Then there can be certain release of chemicals by a leaf damaged by an insect, so that it can start a predatory interaction. Then there can be a production of bitter toxic chemicals that serve as antiferents. Also, there can be release of secondary metabolites by roots into the rhizosphere to attract beneficial soil microorganisms. So you have seen that the secondary metabolites, they may be involved in certain interactions of plants with its environment. They are not involved in the growth and development, but they have been involved in the interaction of plants with its environment. Now we will be seeing how primary metabolites, they act as precursors of secondary metabolites. The primary metabolites are those products which are formed in the end of primary metabolic pathways. those primary metabolites, they act as raw materials or they act as precursors for the production of secondary metabolites. Let us see how that takes place. In front of you, you have a schematic diagram of the different pathways of the production of secondary metabolites, and we can see here the different products of primary metabolism, they are involved in the production of secondary metabolites. For example, on the in the given figure, in the left side, you can see erythrose 4-phosphate and phosphoenol pyruvate. They are arising out of the primary carbon metabolism. Erythrose 4-phosphate is a product of the pentose phosphate pathway. Phosphoenol pyruvate, it is obtained from the steps of glycolysis. These two products combine in the shikimic acid pathway to produce aromatic amino acids, which in turn produce nitrogen containing secondary metabolites. This shikimic acid pathway is also useful for the synthesis of phenolic compounds. In the given figure, on the right hand side, you can see two products. First is pyruvate and the next one is 3-phosphoglycerate. Pyruvate, it is obtained from, it is the end product of glycolysis, and 3-phosphoglycerate, it is a product of the Calvin cycle. These two combine in the methyl erythritol pathway to produce the terpenes. This pyruvate, it also gives rise to acetyl-CoA. Then that acetyl-CoA is useful in the pathway of mevalonic acid pathway. Also, acetyl-CoA, it interns to the proceeding of malonic acid pathway, and other amino acids are also synthesized from this acetyl-CoA. So you can see on the top part of this diagram, the products of primary metabolism are mentioned, and below you can see there is a demarcation of two different colors are given to indicate where the primary metabolism is ending and where the secondary metabolites are formed. So in this way, the products of primary metabolism, they act as precursors for the production of secondary metabolites. Now we'll see the classification of secondary metabolites. The secondary metabolites, it is a large group of compounds and it is divided into three major groups. The first one is terpenes, second group is phenolics, and the third group is nitrogen containing compounds. We will be seeing each of these group of secondary metabolites in detail, how they are synthesized and what are their properties. The first group of compounds of secondary metabolites is the terpenes. The terpenes or terpenoids, it constitutes the largest class of secondary products. These substances of this class, they are generally insoluble in water. They are biosynthesized from acetyl-CoA or glycolytic intermediates, and terpenes, they are formed by the fusion of 5-carbon isoprene products. So 5-carbon product, you can see here the diagram. Yes, this is the basic structure of an isoprene unit. This is one isoprene unit and many multiple isoprene or multiple 5-carbon units join together to form different classes of terpenes. Now terpene synthesis, for terpene synthesis, there are two pathways. First one is mevalonic acid pathway. In this pathway, 3 molecules of acetyl-CoA, they are joined together in a stepwise manner to form mevalonic acid. This mevalonic acid, it is a 6-carbon intermediate which is then pyrophosphorylated, decarboxylated and dehydrated to yield isopentyl diphosphate. This isopentyl diphosphate, it is the 5-carbon isoprene units, and then multiple isoprene units or isomers of this isoprene units, they join together to form different classes or different types of terpenes. The next pathway is methyl erythritol pathway. In this pathway, glyceraldehyde 3-phosphate and 2 carbon atoms derived from pyruvate, they combine to generate an intermediate which is eventually converted to isopentyl pyrophosphate. You can see the pathway here. On the left hand side is the mevalonic acid pathway. You can see that how 3 molecules of acetyl-CoA, they have combined to form mevalonic acid. This mevalonic acid then forms isopentyl diphosphate. On the right hand side, you can see glyceraldehyde 3-phosphate and pyruvate, it is forming a 6-carbon intermediate, which is methyl erythritol phosphate, which then forms dimethyl allophosphate. That is a isomer of isopentyl diphosphate. So these two 5-carbon isoprene products, they join together to form a 10-carbon isoprene or 10-carbon terpenoid, which is known as monoterpene. Also, it is also named as geranyl diphosphate. Similarly, 5 carbon atoms, they join sequentially to form different types of terpenes. Now let us see some important examples of terpenoid compounds. First is pyrethroids or pyrethrins. Now these are a group of chemicals which are obtained from the flowers of chrysanthemum. Now these pyrethrins, they have insecticidal properties and they are useful in the production of commercial insecticide and pesticides. Second is monoterpene of conifers. Now, monoterpene is a type of terpene which is accumulated in the resin ducts of different types of coniferous plants. This monoterpene this monoterpene also has insecticidal properties. Third example of terpenoid compounds is essential oils. Essential oils is basically a mixture of different terpenoid compounds which have a characteristic odor, and they are found in different types of flowers and leaves. These essential oils, they have insecticidal properties, and these essential oils are also useful in the production of commercial perfumes. Also, some of the essential oils, they have medicinal properties. For example, essential oil of lemon, essential oil of mint, rose, etc. Then we have the next compound, that is limonoids. Limonoids, it is a terpene which is a bitter toxic compound found in citrus fruits. This limonite also has toxic effects on insects. Next group is saponin. Saponin is a compound which has soap-like properties. This saponin is useful in the preparation of natural soaps. Now, saponin is widely found in the Retha plant and it is used as a natural soap-like agent. Cardinolates, it is also one example of terpenoid compound which is found in the digitalis plant. This cardinolate, it inhibits the sodium potassium pump, or which the sodium potassium ATPase pump. Last example is rubber. Rubber obtained from rubber plant, it is also an example of terpene. Now next group of secondary metabolites is phenolic compounds. Now plants produce a large variety of secondary products that contain a phenol group, that is an OH group on the aromatic ring. These compounds, they are known as phenolic compounds. Now plant phenolics are a chemically heterogeneous group of nearly 10,000 individual compounds. Some of them are soluble in only organic solvents, while some are water-soluble carboxylic acids and glycosides, and others, they are large insoluble polymers. There are two pathways for the synthesis of phenolic compounds. First is the malonic acid pathway, and the second one is shikimic acid pathway. Now in this diagram, in this schematic diagram, you can see both the pathways. Erythrose 4-phosphate from the pentose phosphate pathway and phosphoenol pyruvate from glycolysis, they both combine to form shikimic acid, which is an intermediate in the shikimic acid pathway. This shikimic acid then gives rise to different aromatic amino acids, and these amino acids then form the different types of phenolic compounds. On the right side, you can see a molecule of acetyl-CoA. It goes into the malonic acid pathway to form different types of phenolic compounds. This is one more schematic diagram to represent the shikimic acid pathway. You can see the different amino acids formed from the intermediate of the shikimic acid pathway, which in turn give rise to the different phenolic compounds. Now let us see some important phenolic compounds. Furanocoumarins. These are some important phenolic compounds which have antimicrobial activity. Then lignin. Lignin, it is an important constituent of plant cell. It is the lignin is the cellular thickening in the plant cells, and it also gives the plant defense against gives. Next compound is lignin. Lignin is an important structural element of plant cells. Lignin protects the plants from herbivore attack. Next group of phenolic compound is flavonoids. Now the flavonoids, they are associated with the accumulation of colored compounds which have UV protecting activity, and also they have antimicrobial and antibacterial activity. Now flavonoids, they are a group of compounds and they are classified into 4 subtypes. The 4 subtypes of flavonoids, they are anthocyanins, flavones, flavonols and isoflavones. This classification, it is based on the differences in the structure of the different types of flavonoids. The anthocyanin, it is the purple colored pigment found in flowers and fruits. This anthocyanin pigment, it helps in attracting pollinators and it helps in seed dispersal. Flavones, flavonols, they are also UV protecting, or they protect the plant from UV damage. And isoflavones, it has antimicrobial activity. Now tannins, it is also one more important class of phenolic compound. It is mostly restricted to woody plants. It is found in the bark of trees, and this tannin, it also has antimicrobial and antibacterial activity. The third class of secondary metabolites which we will be seeing today are the nitrogen containing compounds. A large variety of plant secondary metabolites have nitrogen in their structure. Included in this category are well-known anti-herbivore defense compounds such as alkaloids, glucosinolates and cyanogenic glycosides. Most nitrogenous secondary metabolites are biosynthesized from common amino acids. First class we will see of the nitrogen containing compounds is alkaloids. The alkaloids, they are a large family of more than 15,000 nitrogen containing secondary metabolites found approximately in 20% of vascular plant species. The nitrogen atom in these substances is usually a part of heterocyclic ring, a ring that contains both nitrogen and carbon atoms. They are mostly synthesized by the shikimic acid pathway. In the previous pathway, we have seen how alkaloids were synthesized in the shikimic acid pathway. First, erythrose 4-phosphate and phosphoenol pyruvate, it formed shikimic acid, which then gave rise to aromatic amino acids, and from those aromatic amino acids, different types of alkaloids are synthesized. Now, this is one chart of the different common examples of alkaloids. You can see the different structures of the alkaloids, how they are having a heterocyclic nitrogen in their ring. We can see the biosynthetic precursor, the amino acid from which these alkaloids have been synthesized. Examples of alkaloids are given and what are their uses. Now in these you can see some very important alkaloids present. For example, nicotine, cocaine, atropine, which find their uses in medicinal drugs. Next group of nitrogen containing compounds are the cyanogenic glycosides. These cyanogenic glycosides, they are found in some plants and they release the poison hydrogen cyanide. You can see the reaction here. We have a molecule of cyanogenic glycoside. With the help of enzyme glycosidase, the sugar molecule is cleaved from this cyanogenic glycoside, which results in the formation of cyanohydrin. Then with the action of enzyme hydroxynitrile lyase, or spontaneously also, this cyanohydrin, it can be converted to ketone and hydrogen cyanide. So whenever there is an insect attack, this hydrogen cyanide, it is released so that plant can defend itself from the insect attack or herbivory attack. Next group of nitrogen containing compounds is glucosinolates. This is one more type of nitrogen containing compound which breaks down to release volatile defensive substances. Now these glucosinolates, they are sulfur containing compounds and they are found principally in the Brassicaceae and related plant families. So they are extensively found in cauliflower, radish, and all those members of the Brassicaceae family. You can see the reaction here, how the glucosinolates, they are uh, attacked by the enzyme thioglucosidase to release the glucose molecule, which forms an aglycone. This aglycone can then be spontaneously converted to isothiocyanate and nitrile. So this is also a compound which protects the plant from insects and it defends the plant from herbivory attack or insect attack. One more example, a small group of secondary metabolites are found in the nitrogen containing compounds, which are the non-protein amino acids. These are some unusual type of amino acids and they have their structure similar to that of the common 20 amino acids. Now plants and animals, they incorporate the same 20 amino acids in their proteins. However, many plants, they also contain some unusual amino acids which are known as non-protein amino acids. They are not incorporated into proteins, but they are present in free form and they act as protective substances. The non-protein amino acids, they are very similar to common protein amino acids. You can see the structures here of standard amino acid and the analogous structure of non-protein amino acid. You can see how similar the non-protein amino acid structure is with that of the standard amino acid. For example, you can see the structure of canavanine and arginine. The arginine, it is a standard amino acid, and the canavanine, it is a non-protein amino acid which is having a very similar structure to that of arginine. Second is azatidine 2-carboxylic acid, which is having a very similar structure to that of proline. Proline is also one of the standard amino acid. So these amino acids, they are unusual amino acids which are found in some plants. They can be sometimes incorporated in the process of protein synthesis, but they are not incorporated into the protein. They block this process of protein synthesis and they lead to cell death. Thus, they protect the plants from herbivory attack or insect attack. So, the student assignment for today's lecture, you will describe the role of different secondary metabolites in plant defense and send it to the given mail ID on your screen. Let us see the summary of today's lecture. Today, we have been discussing about secondary metabolites, an important group of plant products which are obtained from the plants as a result of primary metabolism, but they are not involved in the, but they have no role, direct role in plant growth and development. We have seen the different classification of secondary metabolites today. The three major class of secondary metabolites we have discussed, their structures, how they have been synthesized, all that we have seen in today's lecture. In the next lecture, we will be seeing about the qualitative and quantitative study of phytochemicals. We will be studying how the different chemicals or different primary metabolites and secondary metabolites, they are detected in plant, that is their qualitative study. And we will also be seeing how their amount is estimated, how we can quantitatively measure the amount of a particular chemical in a plant. Thank you so much for your time, everyone. We will meet next time.